So you guys might be really excited, but we are done talking about all of the different bacteria. Now we will be talking about some viruses and parasites, so we will be adding more organisms to the list of already a lot of organisms that you will need to know for the final exam. But we're going to deviate in this lecture and talk about antibiotics as well as resistance mechanisms. We're going to start with some definitions. Antimicrobial chemotherapy. This is the use of chemicals to either inhibit the growth of or kill microorganisms in or on the host. Selective toxicity is when an agent that's used must inhibit or kill the microorganisms without harming the host. So this makes sense, right? You want your antibiotics to be able to get inside of a cell and kill that cell. However, since you have to put these chemicals inside your body, you don't want them to kill your own cells. You only want them to kill the bacterial cell. So we'll talk about some of the mechanisms that the antibiotics use to target this selective toxicity to target the bacterial cells and not human eukaryotic cells. There are different classes of antimicrobial agents. There are antibiotics, and these are substances that are produced by usually a living organism. So they're metabolic products from a microorganism that has been shown to either inhibit the growth of or kill another microorganism. So a lot of antibiotics were discovered by accident. There happened to be something else growing on a plate that was inhibiting the growth of what should have been on the plate. Antibiotics are natural products of microorganisms. Now there's also antimicrobial chemotherapeutic chemicals. Now these are antimicrobial chemicals that are synthesized in the lab. So these are man-made antimicrobial agents, whereas antibiotics are natural products that are purified and modified into antimicrobial agents that can be used to treat infection. Now there are different groups of antimicrobial agents. So antimicrobial agents is the safe way because antibiotics are specifically those products that are produced by other microorganisms, and not all antimicrobial agents are actual antibiotics. Some of them are completely man-made. So they are all referred to collectively as antimicrobial agents. There are those that are bacteriostatic, and the bacteriostatic antimicrobial agents inhibit bacterial growth. So it's very important to know they do not kill the organisms, they only inhibit growth. Now, bactericidal antimicrobial agents do kill. So cidal killing, static or stasis to remain steady. There are different types or spectrums of antimicrobial agents. There's broad spectrum and narrow spectrum. So our broad spectrum agents are effective against both gram-positive and gram-negative bacteria, whereas our narrow spectrum agents are effective against only one type of organism. So you might have an antimicrobial agent that will only target gram-positive bacteria. That would be a narrow spectrum antimicrobial agent. Now some examples of broad spectrum will go after both gram-positive and gram-negative bacteria are tetracycline, streptomycin, cephalosporin, ampicillin, and some of the sulfa drugs like sulfonamides. Narrow spectrum antimicrobial agents include penicillin G, erythromycin, clindamycin, and gentamicin. Now, over the years, and I think people working in healthcare, clinicians have gotten much better at not prescribing antibiotics for every little thing. But way back when I was a kid, I can just sniffle. And my mom could call the doctor and say, "Oh, she sniffled. I think she's getting something." And they would just call in a prescription of antibiotics. You took an antibiotic back when I was a kid for any little teeny tiny thing. You didn't have to go in. You didn't have to have any tests done. So what happens with that is antibiotics were overused. So what the results of antibiotic overuse include getting opportunistic infections. So we've talked about some of those, where you take antibiotics and you destroy your normal flora that's supposed to be in your body. You reduce those numbers to a point where you'll allow other organisms that will opportunistically cause infection if they're able to overgrow. So some examples of that are, we talked about it very recently with our ulcerative colitis caused by Clostridium difficile, our anaerobic gram-positive bacillus. So if you reduce your normal intestinal flora, your E. coli and lactobacillus and other intestinal flora, if you're carrying Clostridium difficile, it can overgrow, it can produce toxins, and it could cause that pseudomembranous colitis. Some people also carry yeast in their genital urinary tracts, women especially. So if you take antibiotics and you reduce your normal good flora, your lactobacillus in the vaginal tract, you can end up with a yeast infection. Same thing with Gardnerella vaginalis or Bacteroides, where you reduce your lactobacillus and some of your good flora, and those organisms can overgrow and cause bacterial vaginitis. Now, you can also lead to drug toxicities. You can lead to allergic reactions. There's a lot of people that are allergic to certain antibiotics, and it seems to have gotten increased over the years, people that are allergic to antibiotics like penicillin. And I think the most notorious thing that has happened with the overuse of antibiotics is selecting for resistant microorganisms. So you are infected with some sort of bacteria, you take antibiotics and you don't take them. You're supposed to take them every day for 10 days. You take them for 3 days, you're feeling better, so you stop taking them and you save them. What happens is you might not have killed all of the microorganisms there. You might have killed off enough where you start feeling better. So then you stop taking them, and those that are there have selected for resistance. Maybe they've acquired a plasmid, maybe they acquired a mutation that now allows them to resist being killed by that antibiotic. So the next time you get really sick and you go and take that same antibiotic, all of a sudden it doesn't work any longer. The other, thing is you have the sniffles, you likely have a cold. Antibiotics do not work for viruses. Only antiviral agents will work for viruses, and antiviral agents are only given for certain types of infections, not for the common cold. So again, when I was a kid, you had sniffles, likely a typical cold. You call up and you go get antibiotics for that cold. Well, it's not doing anything for the cold. All it's doing is allowing your bacteria to get hit with this antibiotic, maybe not kill the flora in your body, but allow a mutation to occur where it becomes resistant to that antibiotic. And then when you actually need that antibiotic, it doesn't work anymore, and then you're spreading resistant flora to other people. Now the antibiotic targets and modes of action, and this is where that selective toxicity comes into play. Antibiotics have to target areas that will be toxic to bacteria and not to our own cells. So some of the antimicrobial agent targets are cell wall biosynthesis, folate synthesis, DNA replication, RNA transcription, and mRNA or messenger RNA translation. So here's just a schematic showing all of these different sites that our antimicrobial agents go after. So some of them will go after the structural integrity of the bacterial cell. So they'll go after the cell wall, like some examples are beta-lactam antibiotics or cephalosporins or vancomycin. Some will go after the cell membrane. Now the cell wall is a nice thing to go after in bacteria because our eukaryotic cells don't have a cell wall. So it's specific for our prokaryotic cells. Some antimicrobial agents will interfere with nucleic acid synthesis, such as rifampin that will go after RNA polymerase. Some of them will go after DNA synthesis, like the fluoroquinolones. Some of them will go after the ribosomes, so that you can't get any proteins made that the cell might need. So we're first going to talk about the antimicrobial agents that inhibit cell wall biosynthesis. So these antimicrobial agents will target that bacterial cell wall, which is a wonderful target because it is not going to harm the host cells at all, because the target in the bacterial cell wall is usually peptidoglycan, and peptidoglycan is only present in the prokaryotic cell and not in a human eukaryotic cell. Now, the bacteria have developed resistance mechanisms against some of these antibiotics. So some of the resistance mechanisms that bacteria, now keep in mind these are the bacteria now, bacteria can produce enzymes like beta-lactamase that will destroy the beta-lactam antibiotics. They will produce penicillinase so that you can't use penicillin. They might alter the structure of penicillin binding proteins, so the proteins that penicillin will come in on the surface and bind to, they'll alter those so the penicillin can't recognize those anymore. Now our beta-lactam antibiotics. These antimicrobial agents specifically bind to the penicillin binding proteins, or PBPs, on the cell wall of the bacterial cell. Now there's an active structure in the beta-lactam antibiotics, and this is a 4-membered beta-lactam ring. The beta-lactams interrupt cell wall synthesis and they kill the bacterial cell. The antibiotics that are considered beta-lactams are penicillin, the cephums, the carbapenems, and the monobactams, which these antibiotics can be either narrow or broad spectrum, depending on which agent you're talking about. Now we'll first talk about the penicillins. There is a natural penicillin called penicillin G. This is a narrow spectrum agent because it's effective only against gram-positive bacteria. Penicillin G is inhibited by the enzyme penicillinase. So any organism that produces penicillinase, you would not use penicillin G to treat an infection caused by that gram-positive organism. There are some semi-synthetic penicillins. So penicillin G, natural penicillin, is made from a fungus. It's actually a true antibiotic. But the semi-synthetic penicillins are those that are partially man-made. Those include methicillin and nafcillin. Again, they are narrow spectrum. They are effective only against gram-positive bacteria. However, they're not inactivated by penicillinases. So a bacteria, a gram-positive organism that is producing a penicillinase can still be treated using methicillin or nafcillin. There's also some semi-synthetic broad-spectrum penicillins. These are oxacillin and ampicillin. These are inactivated by penicillinases, but you can use a beta-lactamase inhibitor, and we're going to talk about those a little bit later. So bacteria, we said, can resist some antimicrobial agents such as the penicillins because they produce the enzyme beta-lactamase and penicillinase. If you add a beta-lactamase inhibitor to the antimicrobial agent, you can actually treat the bacteria with that beta-lactam antibiotic with the beta-lactamase inhibitor. So even if the bacteria is producing the enzyme beta-lactamase, it's not going to do anything because there's an inhibitor present. So the beta-lactamase inhibitors include clavulanic acid. That's one of the most common beta-lactamase inhibitors. So sometimes you'll find a combo antimicrobial agent, and clavulanic acid is a lot of times mixed with the beta-lactam antibiotics so that you inhibit any beta-lactamase that might be produced by the bacteria, so you can effectively treat some of those infections. Now, sulbactam is another beta-lactamase inhibitor that is commonly used in conjunction with broad-spectrum penicillins. So sulbactams aren't antimicrobial agents at all. They have no antimicrobial activity. However, they inhibit penicillinase. So if you add it to some of the broad-spectrum penicillins, if you have a bacteria that produces penicillinase, you can still use the broad-spectrum penicillin in conjunction with the sulbactam to treat. The cephalosporins are resistant to penicillinase, so you're not going to treat an infection with an agent with cephalosporin if it is producing a penicillinase. The cephalosporins are broad spectrum. They have been altered over the years. They've expanded greatly, and there's multiple generations of cephalosporins, and each generation has increased its spectrum of activity. So just like everything else, over time they make these antimicrobial agents better and better. So there are first generation cephalosporins such as cephalothin and cephalexin. There's second generation cefoxitin. There's third generation like ceftriaxone and cefotaxime. And there's fourth generation such as cefapime. There's many more generations also. So all the time they're coming up with better and better cephalosporin types of agents. The Carbapenems, an example is imipenem, are resistant to beta-lactamase activity. They're broad spectrum. They inhibit peptidoglycan synthesis. So the peptidoglycan that's in the cell wall of our prokaryotic bacterial cells, they inhibit that from developing, so the bacteria can't survive. Carbapenems are usually combined with cytostatin sodium to prevent degradation of the antibiotic in the kidneys, because some of these antimicrobial agents can be toxic to our organs. Monobactams are broad-spectrum beta-lactam antibiotics, and they are resistant to beta-lactamase producing production by some bacteria. Glycopeptides don't bind to the penicillin binding protein like most beta-lactam antibiotics do. The glycopeptides bind to the cell wall precursors and prevent the development of the bacterial cell wall. So glycopeptides include vancomycin, and the thing about vancomycin is it's huge. It's a very large molecule. So it's only effective against gram-positive bacteria, which makes it narrow spectrum. The molecule is so big it can't get into the cell wall of gram-negative bacteria, so it's only used for gram-positives. We're now going to move on to the second part of our antibiotic lecture.